but you can take it during class time. So generally, it was supposedly for to take it during class time, but letting you take it tomorrow if you want to. Okay, so any questions about the quiz? So it should start around 5. It's available around 5. Okay, the second thing is I posted the review and some research papers, examples of research papers that you can use for your research project, for your research paper. I posted paper, so again, you have two options. You can do a review. There's a rubric for the review. There's a review, or you can do an actual research paper if you'd rather do that. And all of them have to do with cancer and immunology. The other option is a research paper or research article. And so the third thing is, does anybody have any questions about the paper, the research paper?
I have a question. So if we pick a paper, is there just like a sign-in sheet, or how do we reserve this paper so we know that nobody else will work on it?
So you just send me an email.
Mm-hmm. Okay.
Yeah, and then so the other option is you can pick your own research paper. You can have your own paper. I think somebody already has and send me their own paper, and I just want to look at it to make sure it's okay. And so it has to be with anything related to immunology, hopefully immunology and disease or research on immunology. Okay, so you have any of these three options. I haven't done that, but so this paper is due at the end towards the end of April, and so I posted how to do the review, and I need to post how to do the guidelines for the research paper. You do a research paper, so what they call the rubric. So one thing about the review, you just answer the questions that I posted. For the paper, so for the paper, it's so that's the way you set up your research paper when you write a research paper, or review a research paper. Then it's slightly different because the research paper has a lot of data, right? And so you have to follow a different format. And the format is usually you put introduction. So let me start with. So for research, if you do a research journal, or so you call it a research article. So normally what you do is you're going to have the introduction, so the intro. You have to follow a specific format. You have to have intro, then the materials and methods, so M&M. So for materials and methods, the materials and methods is very short. You can just like outline and like the best, you don't have to write paragraphs, so you can just write like a list. You just write a list of the materials that they use. So that would be that. The materials and methods. And then the most important thing usually is the results. And So the other thing is the, the discussion, right? So this is the general format for research paper, and then I'll tell you in the rubric what I'm looking for each one of them. Okay, so I think I misspelled discussion, but anyway, so you have the intro, materials, methods, results, and discussion. And so the other thing about the research paper is that anything, it has to be on your own words, so your own analysis, or any of them have to be in your own words. So you cannot copy, paste, copy and paste from other sources. You can use any source you want. You can use Wikipedia, you can use internet, any sort, but you have to give credit to the whatever source that you got. So you have to cite your sources. And so this is going to go through, you may already be familiar with it. There's a program that checks to see how similar is your paper to something that is already published on the internet. Okay, so that's why you want to write it on your own words. You can be similar, you can be 60, 50% similar to other sources, so that's fine. But your paper cannot be 90 to 100% similar to something that is already, so this is called the Turnitin. And so you cannot copy, like even sections of a graph, of a paragraph, it cannot be 100%. Otherwise, you're going to get points off. So that's considered plagiarism. Okay, so this is the main thing you want to avoid when you write a paper. You want to avoid plagiarizing something. And so you want to give credit to anybody that has already worked on it. And so the best way to get credit is to cite it. So there are different ways to cite something. And again, I'll give you information on how to do it. So you cite the sources. Something that is published, you have to cite. But this is the general outline of a paper. So, any questions?
How would you like us to cite our sources?
The sources, yeah. So I'll give you there are specific ways to, let's say you got something from the internet. So yeah, so I'm gonna give you a rubric. So the rubric is gonna tell you how to do that. So I'm not very picky about how you cite them. So this is not an English class, and you're not gonna publish your paper either. So I'm not very picky about as long as you cite them. Okay, so I'll give you some suggestions on how to cite something. Let's say most people don't read books anymore, so there are ways to cite books. There are ways to cite journals, papers. There are different ways to cite anything you get from the internet, so like a website. And so again, as long as you cite it, I'm not really picky about how you do it. As long as, even if you just put it, I'll take it. Even if you just put it in, so now let's say you copy something and then you say research paper, you copy exactly. Well, you don't want to copy it, but you said your source, this one, you can put it like a one here, an A, and then at the end you can say one. This comes from this link on the internet, or it comes from this journal. You just write the journal. Okay. So as long as you cite it, so again, I'm not going to be picky about how the format, that there is a professional format to, but again, if you were going to publish it, yeah, you have to follow the very strict format. But for me, it's not that strict as long as you cite it. Does this make sense? And so everything, let's say you have a sentence, you write a sentence, and you got the information from an let's say Wikipedia. All you have to do is, this is the first source that you're citing, and so you put one, and at the very end you have a paragraph of sources. So you may have one source, you have two sources, and whatever you have, you're just gonna put sources and then say one. And then you're gonna say here, what did I say? Let's say you got it from Google. And then you put the link. Let's say if it's a link, you just put it here. And that's it. And so as long as it can be traced. Does it make sense?
Okay, yeah, it does.
Yeah. Okay. Be good. So any questions about the paper then? Okay, so kind of next week I want to see if the we can do the ELISA on campus, so on the lab. So I'm going to go over the protocol for the ELISA. I already went over the the background and how it works, and so I'm hoping we can do the ELISA next week and maybe the following week. And so I had to check the materials. I'm going to try to go tomorrow, Friday, and set up, meet with the technician to set up to make sure we have all the conditions. But the other thing I need to know is if you haven't told me yet, then you're going to, there's a few people that are not able to come to the class for whatever reason. They're not able to come to do their labs on campus, and some people do want to do it. And so you're not able to do it on campus, then you can still do it online. And so if I'm on class, let's say we're doing an experiment, uh, I will either take if you want to continue doing it online, I will either take the material that you can access it, or then you're going to be on class. We're not going to be in class all the time. Sometimes we're still going to be online. But when we're doing an experiment, we're going to be on class. And so you had this option, this one or this one. So I need to know though from you if also if you're in a group and some members are able to come and some members are not, but you're willing to share the information with your group, then you're fine too. Um, you they can your roommates can help you do other things like the research paper. Okay, do we have any questions about these? No?
So you're saying that we can either attend online or actual class, or we have the options
Yeah,
to?
well, so there's some, so I want to do an ELISA next week, and I need to know how many people are willing and are going to be able to come to the class to do the ELISA, right? Because if I only have one person, so that's not going to work very good. But also, I don't want to have everybody in the class at the same time. So if everybody's coming, then I can go in the first half, and then the other half can go in the second half and finish the experiment. So I got emails from some people that they said they're not able to come to class at all. And so I'm not going to force you to do that. So that's why I want to know who is able. So you can email me and tell me that, and then so I can set up groups that hopefully, or in somebody in your group, if you already have a group, then you let me know who's able to come.
Do
So
we have to email you to let you know that we're heading to class?
the so oh, let's say your group, you already have a group. You already have a group, and then you can just tell me somebody in your group is coming to the lab to do the lab. Also, when we set up the lab, I want to know how many groups we're going to have. Make sense?
Yeah, makes sense.
Okay, so I hope so. Okay, so now I'm going to go over the ELISA, and then you had the time to take the quiz. Like I said, you can take the quiz today. You can take it tomorrow. It's due tomorrow. And again, so when you answer the quiz, some of them are short answers. So the everything you do when you write a short answer, make sure you write it on your own words. Okay. Okay. So now ELISA. So remember it's been a while since we talked about that. I purify. So let me go back to this slide here. So I purify, or we purified in class last semester. we took rabbit serum and we made a column. And in the column had beads, right? So I'm making the beads bigger than they actually. So this is made out of plastic, and the beads also have a material that are linked to the ligand. And so I pass uh, rabbit rabbit serum, rabbit serum, pass rabbit serum through here, and then we collected what is called the flow through. What the first thing that comes out, we collected, and then subsequently, so the first thing we added rabbit serum. So we collected whatever came through through the serum, whatever was left, whatever didn't bind to this column, we collected. And so we kept, let's say this is 2-1, this is 2-2. And then the second tube, after the serum goes through, then we wash. So we wash with a buffer, right? So we wash with a buffer. And, the wash with a buffer. And we collected these, we call it the wash. And so we collected also the wash. So we do it 2 times. So collected the wash. The buffer here has a little bit of salt. So it has low concentration of salt. And so this is low salt. This is to uh, basically to remember the serum has a lot of proteins. And so when you do a wash, there is some interaction between proteins that are not very strong, they're not very specific. So you can have 2 proteins that interact, but the interaction is not specific. There's some charges that maybe attract to each other, or the concentration of the protein is so high that they're just going to bind to each other. And so you can use low salt, or so you can use low amount of detergents. So usually salt is better because the detergents are hard to get rid of. So you can use detergent. And so you basically what you want, it's only or as much as of your so you have the bead here, and then on top of the bead you have all these your ligand here. So you have the ligand here, and then you're trying to only have the what is left after you wash. You only want your antigen or your IgG, because this is the we were trying to purify the IgG, right? The rabbit IgG. These are the only things we want to have inside the column after we wash. And so we wash. And so in order, this interaction between the IgG and the ligand is very strong. So does anybody, so this is the rabbit IgG. Does anybody remember what the ligand was?
Protein A.
Protein A, very good. So the ligand was protein A. The ligand is, so protein A doesn't come out because protein A is linked covalently to the bead. So this here is the bead. So they're tiny. You can see them actually. They look like grains of salt. So you can see them. So we pour this. You put a filter so that it doesn't come. And then you elute. So you elute with your buffer. Right? So we went over this before. So I eluted. And this one has high salt. And I forgot what it was. I think it was like 3 molar magnesium chloride, I believe. But anyway, so it's very high salt. This is high salt compared to this one over here. So it's high salt. In the high salt, does anybody remember what it does? So these are ions, right? So you have an ion. You have another ion here, chlorine. Does anybody remember what it is really 3 molar is a lot of salt. Does anybody know what this does? What is the purpose of these?
Someone in chat said to separate.
To separate. Yeah, very good. So yeah, it's to separate it to break the bonds, right? So these bonds are very, very tight. The affinity is very strong between the protein A and the IgG, hopefully. And so it's very tight because it's very specific. You can remember the rabbit IgG binds protein A very well. But not all the other IgGs from other animals, the interaction is not as tight. So the charges here compete with the interactions here. So they basically separate. Okay, they separate, and that's why you use a lot of salt. And so once you add the elution buffer, then this is where your protein comes out. And you concentrate it. So you don't add a lot of these, so your protein comes concentrated. So in the end, you have, hopefully, you have your concentrated protein here, your IgG. Okay, so this is IgG. And so this is what I showed you before last time. Remember, I had an SES gels where they have a bunch of bands, and the wash in the serum had a lot of bands, and the one that has the most concentration was albumin, right? And then in the end, I had one that only had one band, and that was my IgG. It was only one band, and so what we did with this one, then we measure the concentration, but we really don't know if the protein I got, this band here, is IgG or not. And so for this reason, we do an ELISA to test if we were able to purify this that we wanted to purify. So this is the purpose of the ELISA. So remember, ELISA is a binding assay, it's a plate assay, and the interaction between your antibodies is going to be very strong. Okay, so this is basically what I did before the ELISA, and so now it's how to do the ELISA. So I hope you can do this. And so for the ELISA, you can put the ligand, so like protein A here, or you can do something easier where you can coat with the serum or 3 animals. So here, remember the serum has a lot of protein. So we basically have 96 well plates. All of these are going to have 1, 2, 3, going to have bovine serum, 4, 5, 6. All of these are going to have sheep serum. And then all of these 7 to 9 have rabbit serum, and then all of these are going to have IgG. This is purified IgG. Okay, so this is different. This is different than this. Okay, so this is serum. This is actual protein. Okay, so what I did with the serum is I used different kinds of serum: bovine, sheep, and rabbit. And in this case, we don't know the concentration of IgGs in serum, right? We don't know because there's so much protein in there. So all of them have IgGs, all of them. So, but they're from different animals. Okay, so why do you think, if I wanted to, the one that I'm testing is this one here, rabbit, I purify rabbit IgG. Why do I want to use different kinds of serum to test? Does anybody have any idea?
Would it be like a comparison of how much of the proteins that's in each species, or?
Yeah, that's one what thing you can do comparison. But we already know that some different animals IgGs bind different to protein A. So it is. You're right. It's a comparison. But what is this called when you compare it to something? Or you're blind, you really don't know. You purify a protein, but you don't know what you have here. So I don't know if this is IgG or not. So this is a question mark, even though the gel looks like it is, because remember it didn't break when I added this disulfide. So I don't know. I'm thinking it's IgG, but I don't know. So in order to make sure that because my IgG may react or may not react, but why are we using different animals serum that bind, they have IgG, but they all bind in different affinities. And so yeah, you're right, one of them is to compare, but what are these used as? What is this going to tell me? And so this is just like the planning stages of doing the ELISA. That's what I'm going. So there's a purpose of adding these, and it tells you something about the experiment. So it's going to tell me if I have IgG or not. Because remember, I already know that the rabbit IgG has rabbit IgG, right? And when I test, would I test if there's an interaction with IgG, I'm going to use an antibody that is specific for the rabbit, right? So the antibody that I'm going to use here is rabbit specific. So it was based on it's an anti-rabbit antibody, anti-IgG actually. So I have an antibody that is anti-IgG, and I know it binds IgG from rabbit. And so why do I want to use these sera from rabbit? What is it going to tell me about the ELISA? If what I'm looking for is to see if I purify these and how well it binds.
We use rabbit IgG. We use the rabbit as like a positive control, right? And it'll just show maybe that there's expression.
Yeah, you are correct. So we are using the rabbit as anti-serum is a positive control. Right. Okay, so if this is a positive control, and I know that any of the different sera bind with different affinities, what are these going to be?
Those are going to be the negative controls because the bovine, we shouldn't see any expression.
Correct. Yeah, very good. These are going to be the negative control because you can have a result where everything turns yellow, right? That means everything reacted with your antibody, and that's not what I'm looking for. I'm looking for to make sure that this one what I purify is actually IgG and binds to the anti-rabbit IgG. And so yeah, this is a positive control. These are the negative controls. So very good. So if everything goes right, what I'm expecting is it's going to be no reaction here and no reaction here. There's going to be some reaction here and some reaction here. So if I don't have these controls, then whatever result I get, I may not be able to, what somebody said, compare to the control. Okay, so in order to test the serum, so the serum has tons of protein. Remember, they're full of immunoglobulins, different immunoglobulins. They're full of pyridoxine. They're full of fibrinogen, not so much fibrinogen, albumin. And so to test them, I use a percent. So I use percent because I don't know I can measure the total amount of protein, but I really don't know the total amount of IgG. I can estimate it, but instead I just use percent. And so when I loaded the serum from bovine, so we have serum. So we take the serum and we dilute it to 1%. So 1%. And then these ones I did serial dilution. So you remember all about serial dilutions now. So this is 1%. And then so 110 dilutions. And so every dilution is going to be 110. So we started with 1% and then 0.1%. So this is, so from 1%, I fill all the wells in this one here. Okay, so 1%. You don't have a lot of protein, you can do 100%, and then you do the next dilution, which is this one, 0.1%. So I put 100 microliters here. So, and then I put another dilution, 0.01 here. And then this one is going to be, so if this one was 0.01, the next one is going to be 0.001. This one, and then the next one is going to be 0.0001. So on until I get to a lot of zeros here. And then the last one usually doesn't have, it's just buffer. So that's buffer, no protein here. And then I do the same with sheep. And so eventually I'm going to have all the wells full of serum, diluted serum from bovine. And let's use a different color for sheep. So let's say this, the sheep, oh, again, so I diluted the sheep serum. And so in reality, they all look the same color, but they have different, they come from different animals, so they're slightly different. And again, all of these you can do because the IgGs bind to protein A with different affinities. Okay, so the first thing you can do is you can and also in this case, they bind to the anti-rabbit IgG with different affinities. Okay, so then I can use rabbit anti-serum here. Okay, so this is one way to do it. So you can coat with the serum, or you can coat all of them with protein A, and then after that, then you add the serum. Then you have the ligand for all of them. Okay, so now you have all these dilutions, and the last one in my case is protein A. I mean IgG, my purified IgG. So here is different. It's not going to be a percent because I know that I quantitated. I used the spectrophotometer to determine the the protein concentration, and I determined that the protein concentration in molarity. So you want to know in molar, so micromolar. And so I calculated that, I converted to micromolar. And so what I did is I diluted in terms of molarity, not percent. So in molarity, this is 1 micromolar. Then I diluted 110, so 110 dilutions again. So that was the reason we did the dilution. So 0.1 micromolar, 0.01 micromolar. And then the next one is this is going to 0.001 micromolar. And so on and on and on until you have like 0.00000. As many dilutions as you can. Okay, so once you do that, then you can add directly the antibody, this case is the in our case was code anti-rabbit. Uh, Anti-rabbit IgG. And so all this means is they took IgG from rabbit and added it to a goat. And so the goat developed antibodies against the IgG from the rabbit. Okay, so it's goat. So it's a antibody that binds very well to rabbit IgG, but it doesn't bind as well to bovine or sheep. So then this one has an AP, so that is going to alkaline phosphatase enzyme that is going to, when you add the substrate, if you have this in there, it's going to turn color. And so the way it works is you're going to have a well. And so we're looking at this is looking at the top, but now looking at, so I turn it around and I'm looking from the side. And so you have a well, you have 100 microliters, and in the beginning you coat with, let's say, with IgG. So the IgG is going to take time, but eventually it's going to find its way to the bottom, and it's going to coat the bottom of the well. So this is IgG. The IgG or the protein that I purified that I hope is IgG. This is rabbit IgG. Rabbit IgG. So I code these, and then the next thing you do, so this will be step number 1. Rabbit IgG. So the next thing you do is, so you look at the surface of this plate. Not all the surface is coated with this protein IgG. So eventually, what you do is you wash your solution that contains IgG or that contains the serum, and you add another solution. And the second solution is going to have a you want to block. So the second solution, you want to have to block these areas here that don't have a protein bound. The most common way to block this is an albumin. So it's albumin, or you can add milk. So the albumin, so basically what you have is you have a protein of albumin. Let's say this is albumin protein. What it's going to do, it's going to bind to the plate, the areas where the antibody is not bound. It's going to cover these, and it's going to prevent any other proteins from binding here. So you're going to have these surface of the plate covered with BSA. Okay, so this is this called a block. You're blocking anything that is. And so you do this because you don't want to prevent the one that is going to react with your IgG, which is in this case is going to be, let's see, I'm going to use a different color. So now I'm going to use green. This is gonna be the goat anti-rabbit. Rabbit. This is over an IgG, and this one has an